Magdalena PLOTCZYK comes from the United Kingdom.
Yes. So I'm a third-year PhD candidate in the lab of Dr. Claire Higgins in the Department of Bioengineering at Imperial College London. So I would like to thank the organizing committee for giving me the opportunity to present today. Yes. So today I'm going to tell you about my research looking at the role of human hair follicles in scar remodeling. So let me first introduce you to hair follicles. And hair follicles are these unique mini organs that undergo constant cycles of growth. And because of that, a single hair follicle can produce many fibers throughout the years. In addition, the surrounding skin is also changing. And the changes include remodeling of collagen and increasing vascularization and thickness of epidermal tissue when the hair follicle is growing. So interestingly, in the case of injury, it's the same mechanisms that are involved in wound healing and are responsible for skin repair. However, in most cases, wound healing results in the formation of a scar. And despite differences in different scar types, all of them are characterized by an abnormal deposition of large amounts of collagen and lack of functional structures such as hair follicles. So now, would it be possible to reverse this process of scar formation by reintroducing a growing hair follicle in order to promote the remodeling changes that we can see in a hair follicle cycle in healthy skin? So in order to answer this question, we've designed a clinical study in a collaboration with a hair transplant surgeon, and we transplanted hair follicles into stretch scars. So these scars are formed in 5% of hair transplantation surgeries when a strip of skin is excised from the back of the head and in order to harvest follicles to be transplanted to the front. So these stretch scars are a great model for our study because they are all formed in the same place, all on the back, on the head. They all are formed in males only, and all because of the same surgical procedure. So in our study, we collected a 3mm biopsy from a scar before transplantation, and then at 2, 4, and 6 months after the transplantation. So we know that a growing hair follicle can induce remodeling changes in the healthy skin, but we don't know whether it can also affect scar tissue. And also, despite many studies being published on scar tissue, most of them are focusing on pathological scarring, such as keloid or hypertrophic scars. So because stretch scars have never been studied before, I've decided to characterize them and compare to the healthy neighboring skin. So we know that there are differences in the composition of extracellular matrix and the network in scarring. So I decided to look at collagen type 1 fibers, and I used second harmonic generation imaging to do that. And in this process, two photons interact with a unique medium, such as collagen type 1, and these fibers have a lack of central symmetry. And thanks to that, photons can interact and combine and create a one second harmonic generation photon that is twice the original frequency and so twice the energy. So this imaging system is great because there is no need for staining, and we can image quite deep into the tissue without compromising resolution. So I can then detect this signal using high sensitivity detectors, and I can scan through quite big areas of the tissue. So after image acquisition, I used a simple 2-channel segmentation to separate the fibers from the background. But I found that it didn't really represent the architecture of collagen. So for that reason, I used an alternative technique, a tool called Elastic. And I trained the software to recognize 3 different features. So there were thick collagen fibers above 10 microns, thin fibers below 10 microns in the background. And Thanks to that, I separated all my images into 3 channels, which represented the architecture much better. So using batch processing on 45 images, I analyzed collagen fibers in both conditions, and I found a significant increase in the collagen density in stretch scars, which was represented by an increase in the total percentage of collagen and lower fiber spacing. In terms of the thickness, there were significantly more thick collagen fibers in stretch scars, while the average thickness of fibers in both categories was significantly higher. I've also looked at the orientation, and I created this color-coded maps of orientation angles, and I plotted the distribution of these angles. So what we can see here is that in scars, the fibers peak at 0 degrees, meaning they all run more or less parallel to the epidermis, while in healthy skin, the angles are much more spread out. So I also quantified the alignment coefficient, which tells us more about the alignment of fibers in relation to each other. So because the previous image analysis can be highly affected by fiber density, I've decided to use another method, and this is based on the intensity of the second harmonic generation signal. And this paper shows really nicely that when all the molecules in the tissue are nicely aligned, this gives us the maximum second harmonic generation signal, while partial alignment gives us much lower values. So I quantified this in my tissue, and I found the intensity of the second harmonic generation signal to be significantly higher in stretch scars, meaning the fibers were more aligned. So I was also able to detect the autofluorescence signal coming from elastin fibers using the same multiphoton system. And I found that the proportion of elastin as compared to collagen type 1 fibers was significantly lower in stretch scars. So now, given that there are differences in the extracellular matrix, I suspected that it might have an effect on the mechanical properties of my tissue. So I used atomic force microscopy, and in this test, we indent a cantilever towards a section of the skin, and we measured the deflection of this cantilever, which tells us more about the stiffness of our tissue. So I compared the stiffness in healthy and stretch scar tissue, and I indeed observed areas of much higher stiffness in the scars. So I also looked at the cellular content of my tissue, and I found that epidermal thickness and dermal cell density were significantly lower in stretch scars. But we know that dermis is very heterogeneous, so in the next step, I'm going to look at the differences in the cell type composition of the two conditions. I was also interested in the level of vascularization, which I found to be significantly lower in stretch scars with no difference in the vessel circumference. And what's interesting here is that this is in opposite to hypertrophic and keloid scars, which can actually show significantly higher vessel density than the healthy skin. So when unfortunately, we cannot see this, but you can come to my poster. So when we look at the epidermal-dermal junction, this characteristic reed ridges that we can see in epidermis will not longer present in scars. Okay, so we know now that there are big differences, structural differences between stretch scars and healthy skin, but I wanted to know how they affect the gene expression. So I collected RNA from the dermis of stretch scars and the dermis of healthy skin, and I've performed an unbiased transcriptomic analysis using a whole genome microarray. So I found 200 genes that were differently and significantly expressed between stretch scars and healthy skin. And we can see an area of 106 genes that were down and 94 genes that were upregulated in stretch scars. So let's look at the cellular distribution of the protein products of these 200 differentially expressed genes. And we can see that there are some transcriptional regulators, there are some cytoplasmic proteins and plasma membrane proteins. But because we know that there are major differences in the extracellular matrix of the stretch scars, I was particularly interested in the proteins deposited outside of the cell. So through literature research, I found 3 proteins that were particularly relevant in wound healing and scarring, and we've heard about them today. So it's thrombospondin 1, thrombospondin 2, and ADAM metallopeptidase with thrombospondin motifs. So I first validated this finding using immunofluorescent staining for thrombospondin 1, and I found a similar trend with downregulation of thrombospondin 1 in stretch scars. And I next isolated fibroblasts from this excised tissue, and I isolated scar fibroblasts, papillary fibroblasts from the top 400 microns of the healthy dermis, and reticular fibroblasts from the lower part of the healthy dermis. So I first tested the expression of my selected targets, and I found a significantly higher expression of thrombospondin 2 in papillary fibroblasts as compared to scar fibroblasts, while the expression of ADAM TS12 was significantly higher in scar fibroblasts as compared to both the healthy fibroblast types. So this data suggests that the phenotype that I observed in vivo is actually partially preserved in fibroblasts that I isolated and cultured in vitro. So in my future experiments, I'm gonna try to look for the extent of this gene expression differences and test the expression of other genes that are regulated downstream by thrombospondins. So I also looked at the behavioral differences of these fibroblast types, and I found that the population doubling time of scar fibroblasts was significantly higher as compared to other fibroblasts, which means that this fibroblast proliferated the slowest. And this can possibly explain the in vivo phenotype of reduced dermal cellularity that I observed in stretch scars. And in order to test the effect on promoting vascularization of different fibroblast types, I collected a conditioned media from them, and I applied the conditioned media to endothelial cells. And I found that there was that scar fibroblasts proliferated significantly lower endothelial cell proliferation than papillary fibroblasts. So once again, this might suggest the link between my in vivo phenotype of reduced reduced density in stretch scars. Okay, so to summarize this part of my talk, so far I showed that there are major structural and gene expression differences between stretch scars and healthy skin, and some of these differences are actually preserved in fibroblasts isolated from the tissue. So this allows me to go back to my original question, which is whether hair follicles can induce remodeling in scars. So I analyzed samples from before transplantation and after transplantation, and I looked at 3 patients with stretch scars. And what's important here is that all the scars were established, meaning they were 4 years old or more, and we assumed that at this point the tissue reached homeostasis. And they were also of a very similar width. So I applied the same collagen analysis pipeline, and I saw no difference in the total percentage of collagen and only a small drop of fiber spacing at 2 months after transplantation. However, when I look at the thickness, there was a significant increase in the proportion of thin collagen fibers after transplantation, while the average thickness of fibers in both categories was also reduced. So this reminds us about a shift towards a healthy collagen phenotype. What I also found was that the fibers became more disorganized with a higher spread of orientation angles at 6 months, which was also supported by a drop in the collagen, in the alignment coefficient. And the remodeling of collagen was also visible looking at the intensity of second harmonic generation signal, which was significantly decreased at 4 and 6 months after hair follicle transplantation, once again showing a shift towards a healthy phenotype. I've also looked at the cellular content, and I look at the cellular content at a minimum distance of 200 microns away from the transplanted follicle. So I wanted to make sure that I'm testing only the host tissue. So I found that the epidermal thickness and dermal cell density significantly increased at 2 months, and they actually stayed upregulated at 6 months, which once again reminds us about the phenotype of the healthy skin. There was also a decrease in vascularization at 4 months with no difference in the vessel circumference. Again, healthy phenotype. So I've also observed a gradual change in the initially flat epidermal-dermal junction with restoration of reed ridges that are characteristic of the healthy skin. Okay, so now we know that when we transplant a hair follicle into a stretch scar, there are major structural changes towards a healthy phenotype, but I wanted to know more about the mechanism by which it happens. So I collected RNA from the dermis of my samples, and I performed another unbiased transcriptomic analysis using a whole genome microarray. And I found over 1,700 genes that were differentially expressed after hair follicle transplantation. And we can see on the heat map, we can see an area of genes that were primarily upregulated and downregulated after the transplant. So having a closer look into these genes, we can see that there were 266 genes that were upregulated after transplantation, and even after 6 months after transplantation, they were still upregulated, and similarly, 294 genes got downregulated and stayed downregulated. Looking at the cellular distribution of the protein products of these differentially expressed genes, we can see a high level of interaction between them. So in my future experiments, I'm planning to look into these interactions in order to understand the molecular mechanism by which the remodeling happens. But another question that remains now is whether this hair follicle-induced remodeling is unique for stretch scars. So I tested that in a case study of a patient with a 50-year-old scar on his thigh, and this scar was completely devoid of hair follicles, and it also resulted from his thigh being a source of a skin graft for another side of the body. So I applied, so first I compared the initial characteristics of stretch scars and skin graft scar, and I found that the epidermal thickness and vessel density were significantly higher in stretch scars. And that could be explained by the proximity of hair follicles surrounding the scar. So we know that scars can be highly heterogeneous and the baseline levels can be very different. But would this also would it also influence the effect of transplanting hair follicles? And to answer that, I've applied the same collagen analysis pipeline, and I found a similar remodeling shift towards a healthy phenotype with a decrease in collagen thickness and higher fiber spacing. So looking at the cellular content, there was also an increase in the epidermal thickness and dermal cell density, as well as vessel density after hair follicle transplantation, once again reminding us about the healthy skin phenotype. So to summarize, in my talk, in my research, I wanted to know whether transplanting of a hair follicle into stretch scars can remodel the tissue and improve its phenotype. But because my model system, stretch scars, haven't been studied before, I decided to first characterize them. And I showed you significant structure and gene expression differences between stretch scars and healthy skin, with some of these differences being preserved in fibroblasts isolated from the tissue. So next, I looked at samples from a scar before transplantation and then after transplantation, and I found that some of the structural differences between scar and healthy skin are actually changing towards a healthy phenotype. So in my future experiments, I'm planning to combine the structural observations and the transcriptional analysis in order to identify the biological processes and the underlying molecular mechanism by which a hair follicle can induce remodeling in scars. And hopefully, in the long term, the results of this study will allow me to identify the target, which we can use to design therapeutic strategies to reduce scarring. So I would like to thank my supervisor, Dr. Claire Higgins, who's here in the middle, my whole lab, the imaging facility at Imperial, our clinical collaborators, and my sources of funding. Thank you.
Thank you very much, Magdalena. Do you have questions to the speaker, to the presenter? Well, think about this, and I'll ask the first question. The density of hair follicles that you graft is always the same.
Yes, it was also always the same, and it was reflecting the same density that would be used for hair transplantation surgeries.
So the influence that you suspect for the modification and reversal of scarring, it does come from the grafted cells, probably.
Yes, definitely. So hopefully by investigating the different targets that I found in the microarray, I'll be able to translate this into the cell-based experiments, and in a year I will tell you exactly which cells and how would be contributing to the observed effect.
Thank you. Dr. Marty has a question for you.
So, very nice presentation, thank you, very stimulating. I was wondering if you could consider that air grafting, I don't really know how it's performed, but can air grafting be considered as a wound itself, a new wound that you create in the previous wound somehow, and can it contribute to trigger a remodeling response in this way, or is it just...
Yes, so that's a great question. So there is a procedure called microneedling, which is claimed to improve scar phenotype or appearance of the scars. However, unfortunately, there is not much literature concerning the molecular effect of microneedling. But if I can refer to some of the previous studies. So in this study, a clinical study compared the effect of hairy punch grafts with non-hairy punch grafts, and the author showed that the hairy punch grafts promoted wound healing better than the non-hairy punch grafts. So we believe that like the hair follicles are actually the active ingredients that can change the skin. And that's something I'm gonna check using fibroblasts isolated both from the skin and from the hair follicle in order to test how potent they are in remodeling and changing the skin.
Yep. There are no... Yes, Lionel? Last question, please.
Thank you for your talk, and congratulations. So, just to go back to the genes that you screen across this method, so did you find some special thing around repressive genes that could be controlled by this kind of approach? I'm thinking, for example, of myelin or something like that. Do you check this type of gene?
Could you repeat the question?
Myelin. M-E-A-N-D-E-N. Myelin. You know?
Myelin?
Yeah.
Oh, myelin for myelination.
Myelin, like the repressive gene in wounds described by people from the Gallo Labs and the Scripps Institute in San Diego, for example.
I mean, it's an interesting suggestion, but I haven't come across any genes like that in my lists. But I'm definitely looking closer into the different possible mechanisms which is almost a never-ending list of possibilities.
Yes, because it could be an option in the future to create.
Yeah, so definitely, I'm looking for suggestions, so thank you for this.
Thanks a lot. Thank you, Magdalena.
Thank you.
And the next speaker would be